everyone, I'm Apoorva Abikar, 4th year graduate student at Center for Human Genetics, Bangalore. I'm grateful for the opportunity to be applying for this award. Today, I'm here to give a short overview about our work. Our lab mainly focuses on understanding chemoresistance in different kinds of cancer. For my graduate study, I chose to understand what is the mechanisms driving castrate resistance or endocrine resistance in prostate cancer. There are many reasons that have been reported in the literature. One among those is the presence of androgen receptor variants. These variants have the ability to interact with the other proliferative pathways or to activate the different sets of transcriptional genes and also to activate the non-genomic actions of androgen receptor wild type gene. So we wanted to understand what is the exact mechanisms driven by these variants. So we treated our variant expressing cells with the drug called as flutamide. This drug is commonly used to treat prostate cancer. So what we observed is that the drug did not have any effect on the variant expressing cells, and we also observed during the course of therapy there was increase in the expression of these variants. So we hypothesized that this variance emerges and their expression increases as a person moving from sensitive to the resistant phase. And also we observed increased cell proliferation in the variant expressing cells. So we suspected the increased cell proliferation may be because these variants are interacting with the other proliferative pathways and they are contributing for the proliferation. To prove this, we inhibited one of the proliferative pathway and we observed significant decrease in the cell proliferation in the variant expressing cells when compared to the wild type expressing cells. So by all this observation, what we concluded that it becomes very important to look for this variants and also their interacting partners when you are treating prostate cancer, especially with the endocrine therapy. Prostate is a gland which mainly depends upon the androgens for their growth and development. When a person is diagnosed with a prostate cancer, clinicians will advise him to administer estrogen as a course of therapy to achieve chemical castration. So we wanted to see what is the effect of this estrogen on the prostate cells. When we treated our cells with the estrogen, we observed increase in the cell proliferation. So we suspected that this increase in the cell proliferation may be because of the activation of estrogen signaling. So we are working on the same to understand what is the exact mechanisms underlying their contribution to their cell proliferation. So therefore, it becomes extremely important to look for the opposite sex hormones and to maintain and to understand the signaling in the different sex. So in the end, what we are trying to tell is that it becomes extremely important to keep all these aspects in the mind when you are treating the hormonal cancers, especially with endocrine therapy. Thank you.